Welcome to the first semester of Human Anatomy and Physiology with Dr. Jason Laughlin, a course that considers the basic principles of human biology, including the chemistry important for biology, cytology, biological energetics, cellular reproduction, DNA, tissues, bones, and muscles. If you have a question for Dr. Laughlin, be sure to post it on the course discussion board. Now, here's Dr. Laughlin.
Okay, the special senses. In this specific lecture, we're going to talk about olfaction and gustation, that is, smell and taste. When we talk about sensation, we know that sensory receptors vary greatly. Some are very simple and some are very complex in terms of the anatomy of the actual receptor. There are two major categories that we can put any type of sense in, and those are somatic versus special. So somatic senses, these are going to be associated with receptors for touch, pressure, temperature, and pain. And there's a handful of other things as well that go there. But these receptors are widely distributed throughout the body. They're found deep in the skin, deep in tissues, and structurally they're very simple. So they're like a free nerve ending or a Meissner's corpuscle, something like that. In contrast, the special senses, here these are receptors that are associated with smell, taste, hearing, balance, and vision. And these receptors are much more complex. They are specialized sensory organs. They are not just simple receptors. And these receptors are within large, complex sensory organs, always found in the head. Okay, so big difference between special senses and the somatic senses in terms of the complexity of the receptor as well as the location and the distribution of these receptors. Well, let's talk about the first one, the sense of smell. This is known as olfaction. The nose contains about 10 million receptors for the sense of smell. And all of those receptors, all 10 million of them, are found in a relatively small area up in the superior part of the nasal cavity. That part only is about 1 square inch in surface area. It's found in the upper part of the nasal cavity, just below the cribriform plate of the skull. And you'll notice here, this is the area here where the cribriform plate would be located. In the area of interest where we find the receptors, they are referred to as the olfactory epithelium. And when we zoom in on the olfactory epithelium, we see here, first of all, is the olfactory bulb that we've seen before, and there's the cribriform plate. But when we look at the olfactory epithelium, there are 3 major types of cells that we see here. The first type is referred to as an olfactory receptor, or as your book calls it here, an olfactory sensory neuron. These are neurons that begin here, and they're bipolar neurons. And so this is going to be the beginning of the olfactory pathway, which we'll talk about in just a second. But they're bipolar neurons, and they have their exposed tips that form these knob-shaped dendrites down here, found in a mucus. They have to be covered in a mucus because the molecules that we are able to detect for the sense of smell have to be dissolved in a fluid, because this is a chemical sense. This is a type of sense that's chemical related. Chemically related meaning that it has to be dissolved in some type of fluid. And for the sense of smell, that fluid is mucus. So any kind of chemical that we can detect through the sense of smell is referred to as an odorant. And odorants are going to stimulate these olfactory epithelial cells here, these olfactory cilia found here at the very ends of the dendrites of these bipolar neurons. That's where the odorant molecules have to bind. And we'll talk about that stimulation here in just a second. The other types of cells that we find in the olfactory epithelium are the supporting cells. These are essentially pseudostratified columnar epithelial cells that are found in the mucous membrane here in the nasal cavity. And they're going to do a couple of things. They're going to provide physical support, nourishment, and they're going to provide electrical insulation for these olfactory receptors. They also help to detoxify some of the chemicals that come into contact, some harmful chemicals that come into contact with the olfactory epithelium. And the third type of cell that we find here in the olfactory epithelium are referred to as the basal stem cells, or as your book is calling it, the olfactory stem cells. These are found between the bases of the supporting cells, and these continually divide to produce new olfactory receptors. These olfactory receptors only live about a month, 30 days or so, before they're being replaced, and they're replaced by these olfactory stem cells. Now, this is actually a pretty remarkable process because most neurons, as we know, once they're mature, they're not replaced. They can't be replaced if they're damaged because most have no stem cells present. But here in the nasal cavity, we do have olfactory stem cells or basal stem cells here that do replace those dying neurons. Now, also found here in the olfactory epithelium are the ducts, the ducts, the ducts of these olfactory glands that are found up here in the connective tissues, the lamina propria, just above the olfactory epithelium. These olfactory glands are sometimes called Bowman's glands. These are exocrine glands that produce mucus that's carried to the surface of that epithelium by these ducts. So both of these olfactory glands here and the supporting cells of the epithelium, they're controlled by branches of the facial nerve. Okay, facial nerve regulates these or controls these glands. And so when the facial nerve needs to produce a lot of mucus, maybe something irritating has hit the eye or irritating has hit the nasal cavity, the facial nerve will send an impulse to the olfactory gland as well as to these supporting cells to produce more mucus to flush out that material. Now, in terms of the olfactory pathway, we want to talk a little bit about how how these odorant molecules stimulate these olfactory receptors. What is it that they do to stimulate them? And then what direction or where do they go once they're stimulated? So the olfactory pathway really begins at those olfactory hairs that we mentioned. And if you go to those olfactory hairs and you go into the membranes of those olfactory hair cells, what you see is something we just mentioned a few minutes ago, a few lectures ago, and that is G proteins. The olfactory pathway is initiated by G protein. So here we have an integral protein found in this phospholipid bilayer of the hair cell, or the olfactory hair. Here we have an odorant, and the odorant is going to bind to that receptor. That receptor has a specific binding shape. Only that odorant molecule can stick there. And when it does, it's going to activate the G proteins. So this is a highly abbreviated G protein illustration from your textbook. But you'll see here that this G protein is going to influence adenylate cyclase. And you'll recall adenylate cyclase, once that happens, takes ATP, breaks down 2 phosphates from it, and you get a CAMP, cyclic adenosine monophosphate. That CAMP in this case is going to open up open up an ion channel, which is going to allow sodium and some calcium to diffuse through, which will then depolarize this cell. So when that happens, when you activate the G protein network, you open up those channels, and that's going to get this pathway started at these olfactory cilia cells here, these olfactory cilia of the olfactory neuron. You depolarize that here, it's going to then propagate along the bipolar neuron through the cribriform plate, and it's going to form a synapse up here in the olfactory bulb. And those cells are going to take the electrical signal to the olfactory cortex. The olfactory cortex is where that sensory data is going to go for interpretation. So there are 2 major pathways that take information to the olfactory cortex. One pathway brings the information to the frontal lobe, just below the orbit. Okay, that's where smells are going to consciously be perceived and interpreted. So just identification of the smell. The other pathway is going to go to the hypothalamus, the amygdala, and parts of the limbic lobe. And that's where your emotional response to that odor is going to occur. So when you have if you haven't smelled something for 20 years, 25 years, and then that smell comes back, you have that emotional response to that smell, as well as being able to identify it. And sometimes it's easier to emotionally remember the smell and remember some emotional response to it, and then finally you'll be able to identify it. So they go hand in hand, but they are actually interpreted in different parts of the brain. Okay, let's talk a little bit about gustation, taste. Like smell, taste is also a chemical sense. That means the stimulating molecule or the chemical that initiates taste, which is called a tastant, has to be dissolved in some kind of fluid, of course, which is saliva. But remember, odorants are what stimulates the olfactory system. Tastants stimulate the gustatory system. And although that's not exactly always the case, they can overlap, and we'll show you that in just a second. But for a taste receptor to be able to respond to the presence of a chemical that you can taste, it has to be dissolved, and it is called a tastant. Now, taste is actually a lot simpler than olfaction. Didn't mention it, but olfaction, there's about 4,000 different odors that most people on average can identify. Taste, there's only 5 classes that can be distinguished. 5 classes of taste that can be distinguished. Those 5 classes are sour, sweet, bitter, salty, and a relatively new one called umami, which is a meaty flavor or a meaty taste. And flavors in general, when we talk about flavors, there's a lot of flavors like chocolates and pepper, things like that we can detect, but that's a combination of these 5 major classes of taste. Now, odor from food that you actually put into your mouth is probably more important than the actual taste itself. The foods that you put into the oral cavity here propagate up to the back of the oropharynx and into the nasal cavity, and there they stimulate the olfactory receptors. And because olfaction is more sensitive than taste, for example, a given odor may stimulate the olfactory system 1,000 times more strongly than a stimulus from the gustatory system. And so consequently, when someone has a cold or allergies and they complain that they can't taste their food, what they're really complaining about is that they can't smell their food because the mucus builds up in the nasal cavity during that time, and it blocks the odorants from getting into that mucus, deep into that mucus where the olfactory receptors are located. So, when you have a cold or you're stuffy and you can't taste your food much, what you're really not able to do is to smell that food, and that olfactory system is a lot more sensitive than the gustatory system. Now, in terms of where the receptors are found on structures called taste buds, and most people have about 10,000 taste buds, most of which are found on the tongue, but you also have some on the soft palate, you have some on the pharynx and the epiglottis. And the number of these taste buds does decrease with age. There are several types of cells that we see here. Most taste buds are going to be oval shaped like this one here. And you can see the neuron here that is synapsing with these receptors. But there are 2 major types of cells that your book identifies. One is a gustatory epithelial cell here. Sometimes these are called supporting cells / gustatory receptors. But some of these you'll notice have these gustatory hair cells. These gustatory hairs, these are stereocilia, just like we saw back in the gustatory pathway or the olfactory pathway. But these gustatory hairs, these stereocilia, they're actually protected pretty well by this taste pore. I'll show you from a wider angle in just a minute of what we're actually seeing here. But there's a lot of protection of these taste buds in these taste pores. But in terms of what these 2 cells do, the basal stem cells, same thing. They're basal epithelial cells. They're going to replace these gustatory epithelial cells through time. Gustatory receptors only live about 10 days. About 10 days on the gustatory receptors. Olfactory receptors, remember, live about 30 days. So these have to be constantly replaced as well. But on the gustatory epithelial cell, that's the cell that actually responds to the tastant. And these neurons, notice that the gustatory epithelial cells are not neurons. When they are stimulated, they release a neurotransmitter which then stimulates the neuron right here. These are the dendrites of the neuron, and they're looking for stimulation of those gustatory receptors. So 3 types of cells, 2 types of cells found there. And in terms of where you find these taste buds on the tongue, when we're looking specifically at the tongue, they're found on structures called papillae. And there are 3 major types of papillae where we find taste buds. This first one, where we find the most taste buds, are on valate papillae, or sometimes called circumvallate papillae. And they're in the back here of the tongue. They kind of form this V-shaped structure on the back of the tongue. Each one of these has about 100 to 300 taste buds. Another type, the fungiform papillae. These are mushroom-like papillae, mushroom-shaped elevations that are scattered all over the surface of the tongue, and each one has about 5 taste buds each. And then there are the foliate papillae. These are on the side of the tongue. Foliate means leaf-like, and they kind of have the venation pattern of a leaf, located on the trenches of the lateral aspect of the tongue. And most of these actually break down. They degenerate in early childhood. Adults don't even have them. And then there's another type of papillae here called a filiform papillae, which is not even labeled on this slide because they don't have taste buds, but they're all over the tongue, and they just give the tongue a little bit of surface, a little bit of texture on the tongue. On this valate papilla here, also known as a circumvallate papillae, I just wanted to show you this slide because the taste buds, very, very sensitive. You don't want the abrasive food matter breaking those stereocilia, those hair cells. So they're fairly well protected in these pores. So the tasted molecules go down here, and then they stimulate those taste buds. So they're very well protected. But in terms of how we get this receptors, how we stimulate, how do taste stimulate these receptors? There's actually a couple different ways that they can possibly do it. And it really depends on the type of taste that we're talking about. So let's see if I can sketch this out really quickly. Just a real quick sketch here. Okay, so in this particular system, the way that this works is if it is a salt, so salty things, salty things would be like sodium, right? If you eat a lot of sodium chloride, a lot of salt, you have a lot of sodium on the outside of the cell in the saliva. Those sodium cations are just going to simply diffuse through sodium channels. And when they all start coming in, because you have a lot of them on the outside of the cell, that's going to depolarize this membrane, right? So salty foods do that. Acidic foods also, same thing, acidic foods. Instead of sodium, it would be things like hydrogen ions. So there'd be a lot of hydrogen ions out here that are going through these channel proteins. But in either one of those, if it's a salty or a sour, I should have said sour, not acidic, same thing. Sour foods. Sour foods have a lot of acid in them. And so that's going to depolarize that receptor. All right. It's going to depolarize that hair receptor. In contrast, if it is a sweet, if it's sweet, let's change the color here. Sweet. Bitter. Or umami. Okay. In this case, if it's one of these types of receptors, so let's say we have a sugar, glucose here. This is glucose. You've just eaten a lot of sugar, and these glucose molecules are going to bind to the receptor. And in this case, what's going to happen is notice that there's no channel here, but there's G proteins. There's our gamma, the beta, and the alpha. All right. And then there's different ways that these G proteins, as we've talked about before, they may open channels somewhere else. They may influence transcription translation, or they may influence the activity, for example, might activate intracellular enzymes. But it's going to stimulate G proteins when you bring in these particular tastants. Now, there are 3 cranial nerves that are involved in propagating that information to the olfactory or the gustatory cortex. There's the facial nerve, which is going to serve the anterior 2/3 of the tongue. The glossopharyngeal nerve serves the posterior 1/3 of the tongue. And then the vagus nerve serves the throat and the epiglottis. Once those taste buds are stimulated in these areas, those nerve impulses propagate along these particular cranial nerves to the medulla oblongata. And from there, Once it gets to the medulla, from there, most of those axons actually project that information to the thalamus. And then from the thalamus, it then goes to the taste area of the cortex, which is in the insula of Ryle. So here we go with an illustration. You can see here's where the vagus nerve is taking that information from the epiglottis and the throat. And you can see here where the glossopharyngeal nerve is taking it from the posterior 1/3 of the tongue and the facial nerve from the anterior 2/3 of the tongue. If tastins are stimulating those parts of the tongue, these cranial nerves take it to the medulla oblongata. Specifically, there's telling you here the solitary nucleus in the medulla oblongata. From there, it propagates up to the thalamus and then to the taste cortex, which is in the insula, or the insula of Ryle. Okay, I think that's smell and taste. We'll talk about vision up next.
Welcome to the first semester of Human Anatomy and Physiology with Dr. Jason Laughlin, a course that considers the basic principles of human biology, including the chemistry important for biology, cytology, biological energetics, cellular reproduction, DNA, tissues, bones, and muscles. If you have a question for Dr. Laughlin, be sure to post it on the course discussion board. Now, here's Dr. Laughlin. Welcome to the first semester of Human Anatomy and Physiology with Dr. Jason Laughlin, a course that considers the basic principles of human biology, including the chemistry important for biology, cytology, biological energetics, cellular reproduction, DNA, tissues, bones, and muscles. If you have a question for Dr. Laughlin, be sure to post it on the course discussion board. Now, here's Dr. Laughlin.